Welcome, everyone. Thank you all for joining us today for ISB's Research Roundtable. This is a series of talks to keep you up to date on our research. Our intention is to have one of these just about every month through the end of the year, so keep an eye out for our emails. Today, we'll be hearing from our featured scientist. Then there will be an opportunity for questions and answers after that. There's a Q&A box at the bottom of your screen, and you are welcome to submit questions at any time. I'll be monitoring those, and when Dr. Roach is done with his presentation, we'll move to the question and answer session and we'll work through those questions. And today we have a giveaway. We are giving away a signed copy of Temple Grandin's new book, The Outdoor Scientist. Dr. Grandin is a scientist, inventor, and animal activist, and she'll be in conversation with ISB's president, Jim Heath, at Town Hall Seattle on June 10th. And we hope that you're able to join us for that virtual event. We will announce our randomly chosen winner at the end of today's program. And just by being on this call, you're entered to win. Now I'd like to introduce another member of the ISB family, Annie Delucky. Annie is a valued member of ISB's foundation board. She's worked in the Seattle hospitality industry for over 25 years. She currently serves as the president of the Concierge Guild of Seattle, and she serves on the board of Holy Names Academy. Annie is one of ISB's biggest cheerleaders, so we're very happy to have her introduce our featured speaker today.
Thank you so much, Renee. I have the pleasure of introducing Dr. Jared Roach, Senior Research Scientist at ISB. If you joined us for our last research roundtable, you heard Lee Hood refer to the clinical trials we're doing with Alzheimer's. Tonight, Jared will be sharing the details of one of those trials. Jared is a scientific innovator. He was a leader of the first team to sequence the whole genomes of a human family, demonstrating family sequencing as an approach to find the cause of rare genetic diseases. This clinical methodology has become one of the fastest technologies to ever move from research proof of principle to worldwide clinical adoption as a standard of practice, becoming commonplace less than 5 years after ISB's initial publication. In his spare time, Jared has been volunteering to give vaccines at Lumen Field. Welcome, Jared.
Thank you. So, I'll be talking about a clinical trial at ISB today, the Coaching for Cognition in Alzheimer's trial. So Alzheimer's disease is a really important disease. It affects millions of people in the United States and worldwide. About 1 in 10 men and 1 in 5 women will get Alzheimer's disease at some point in their lifetime. If you average the risk of death from Alzheimer's for all Americans over the last year and compare it to the average risk of COVID, the risk of dying from Alzheimer's was about 1/4 the risk of dying from COVID. So very comparably severe diseases in terms of their public health mortality in the United States, even so that people certainly a year ago, and I've even seen these arguments today, would sort of say, well, you know, we really shouldn't do anything about COVID because we don't do anything about Alzheimer's, and that kills almost as many people. And so personally, I've actually find that logic somewhat compelling, but sort of in the opposite way. It's not so much that we shouldn't be doing a lot about COVID, but that we should be as passionate about curing Alzheimer's and treating Alzheimer's as we have been about COVID over this last year. So ISB has been very interested in Alzheimer's because of its importance to public health, but also for another reason. Alzheimer's is a very complex disease, and systems biology is drawn to complexity. So we're really intellectually excited by Alzheimer's and the complexity of Alzheimer's, and we really like doing something that's got potentially a great public health benefit. So how did we get to picking the particular clinical trial that I will describe today as our approach to Alzheimer's? So currently, we have 3 clinical Alzheimer's studies. So we're developing a portfolio of Alzheimer's projects, and ISB is also doing a number of clinical trials in other areas. And what I'll talk about today is our first clinical trial, the COCO trial, which started in early 2018 and will end next week. The COCO trial is a prospective randomized controlled trial to test a particular hypothesis. And that hypothesis is that we can take individuals who have early onset Alzheimer's, a diagnosis of Alzheimer's, but still without too much degradation in their thought process or functional abilities, and treat them with a telephonic coached intervention to improve mostly non-pharmacological interventions of using diet, changes in diet, physical exercise, and brain training to see if we can improve their Alzheimer's compared to controls. So to develop why we're doing this, let me step back a little bit. So around 1900, a gentleman named Oscar Fischer and a number of his colleagues, they were pathologists, noticed in the brains of many people with dementia, these little neurofibrillary neurofibrillary tangles. And they described this syndrome of dementia that associated with these neurofibrillary tangles. So Oscar Fischer is person A, and one of his colleagues, Alzheimer's, is person B in this slide. Alzheimer's wrote a clinical study of person C. His name was August Dieter, who had dementia, and on autopsy, his brain had these plaques. And so this clinical synopsis written by Alzheimer's became somewhat famous. And 10 years later, around 1910, neurologists named this disease after Dr. Alzheimer's, much to Oscar Fischer's chagrin. Neither Fischer nor Alzheimer's made the claim that these plaques, that the amyloid protein in these plaques cause Alzheimer's disease. But by the middle of the 20th century, it was universally accepted, but without much evidence, that the amyloid plaques caused Alzheimer's disease. And that became known as the amyloid hypothesis. So by the end of the 20th century, nearing the end of the 20th century, a young Dr. Roach, I was not yet a doctor, like many others, sort of assumed that the cause of, was because I was trained in this field, that amyloid caused Alzheimer's disease. And I would daydream, like I daydream about hovercraft and superheroes, that I might be able to magically cure Alzheimer's disease if I had some magic wand that could erase these amyloid from the brains of patients with Alzheimer's, and then their cognition would improve and they would get better. But I decided I didn't know how to make such a magic wand. So instead I went into systems biology. But it turns out that the big pharma companies around the turn of the 20th century, 100 years after Alzheimer's, came up with, indeed, such a magic wand, the anti-amyloid pharmacologic therapies. And they were able to do very impressive clinical trials where they tested these amyloid therapies, removed amyloid from the brain, and they saw no change. So patients did not improve their cognition, effectively disproving this 100-year-old theory that amyloid caused Alzheimer's disease. This was wonderful news for Alzheimer's. It seems like it's not wonderful news, but it really was wonderful news because it suddenly freed the field of the stagnation of imagination caused by this dominant theory and allowed all kinds of research, some of whom were not neurologists, to come up with new ideas for how Alzheimer's might be caused and how there might be other effective treatments for Alzheimer's. So really in the last decade, particularly in the last 5 years, we've seen a real flowering of ideas. And it's really a result of these new ideas that has led us to pick some of them to include in our clinical trial. And I'll mention just a couple important elements that drove us in this direction, coming from opposite sides of the world. So one was a Finnish trial, the Finger trial. This was the Finnish geriatric trial. It was not done on Alzheimer's patients. It was done on aging individuals to see if treatment with sort of a top-down idea, where people had done epidemiology and noticed that in populations around the world, people who got more exercise tended to be cognitively better over time. People that ate better diets tended to do cognitively better, and people that did brain training tended to do better. So they combined all three of these into a multimodal intervention and showed that over short periods of time, as well as over a decade, that this Finnish population who was treated with this combination therapy did better cognitively than people who were not. So that's sort of idea one. That says, well, maybe I should try that on people with Alzheimer's disease. Maybe it'll work there as well. In addition to the sort of top-down epidemiologic thinking, other scientists, including a fellow named Dale Bredesen in California, started to think from bottom up. And Bredesen's training was in the molecular biology of neurons. And so he's thinking, well, what causes dementia? Sort of, it's a failure of the brain. And what's the most important physiological part of the brain? It's these synaptic junctions where neurons talk to each other. And he knew a lot about synaptic junctions. And so he's thinking, let me list everything I can that I can think of that improves synaptic function, as well as make another list of everything that degrades synaptic function. And that's largely a molecular list. But then he stepped back and said, now let me think about interventions I can do to humans as a whole, either pharmacologic or not, that improve or stop the degradation of synapses. And his list of interventions included, very similarly, some of the same interventions that were used in the finger trial. So we get sort of both from a top-down and a bottom-up approach. There might be ways of non-pharmacological ways of treating Alzheimer's disease. And this was sort of rooted into people thinking about, well, maybe there's multiple causes, such as toxicity, inflammation, other things that affect synapses or are otherwise observed to improve cognition in aging people. And then for each of those, we can think about ways of intervening. And so the COCO trial is our attempt to synthesize some of these ideas and operationalize them in a way that is logistically feasible. So we didn't do all of these interventions in the COCO trial. We did those interventions that we thought we could convey well using health coaching. Now, there's a certain argument that says that for people who have read all of this literature and have followed the finger trial and have seen all the relevant episodes of Dr. Oz about healthy living, they're convinced, right? And to be honest with you, if you ask me personally, those Dr. Oz shows and all the literature convinces me too. If I had Alzheimer's, I would do these things even if I never saw a clinical trial. The problem is there are millions of people out there with Alzheimer's, and most of them are not convinced. And clinicians in general are not convinced. They're skeptics out there. So if this is truly an effective method and we truly want it to benefit the world, we need to provide more convincing data that this is a reasonable approach. But it's more than that. Even though I'm convinced that this general approach works, I actually don't have a really good sense of what the best approach is. I don't just want to sort of do the current best guess at how to treat Alzheimer's disease. I want to understand how it works, and I want to improve it, and I want to personalize it so that we can give the best treatment to each individual. And we're certainly not there today. So one of the goals of our trial here at ISB is not just to test this simple hypothesis, but is to create a starting point for continuous process improvement, where we do series of trials, with each trial learning not only more about Alzheimer's disease, but also how to improve the therapy each time. Now, let me step back a little bit and explain why there's skeptics out there, for those of you who are not skeptics. So some of the pieces of evidence that are presented today that we have that this might be a reasonable way to treat Alzheimer's disease is that in a number of clinics, including Dale's clinic, including clinics at the Brain Health Research Institute led by Mary Kay Ross, who works with ISB, we've seen patients come in with Alzheimer's disease, do this multimodal intervention and improve or get much better. In some cases, their cognition goes up. In some cases, their function dramatically improves. And so we're really impressed. The question is this a miracle? Well, it's sort of a miracle, no matter how you look at it, but what kind of miracle is it? Are we talking walking on water, or are we talking about getting the kids to school on time kind of miracle? And to address that, one of the things we can do is look at what's already known about the natural history of Alzheimer's disease. So there's been a study by a consortium called the Alzheimer's Disease Neuroimaging Initiative, where they looked at people with Alzheimer's disease over a period of about a decade and measured many, many different things on them. And I could make the point I'm about to make using their radiology imaging or other scores. I'll make it using the MoCA test. The MoCA test is most famous recently because Donald Trump had a perfect MoCA score. So he famously claimed to have a MoCA of 30. So looking at the ADNI data, the people that started with a diagnosis of Alzheimer's ended up losing about 1 point of MoCA every year with Alzheimer's disease. So we can expect anyone with Alzheimer's disease to slowly lose, on average, their MoCA score. So if Donald somehow ended up with a diagnosis of Alzheimer's disease, we would imagine that after a year later, if I had to guess, his MoCA would drop by about a point and become 29. So that doesn't tell us whether or not anybody can ever improve, right? That's average. What we're interested in is individual trajectory. So if I take exactly these same points and just connect them by the individual who they were measured in, we can see that some individuals actually do increase their score at least transiently. It's a little hard to see the maze of data and exactly how many of them go up. But if I sort of summarize the data from the last slide, you can see that after a year, about 22% of Alzheimer's disease patients will actually improve their cognition, at least by this test, and by many other measures as well. After 2 years, the disease is a little bit more inexorable, but still about 5% of them will improve. So when we see stories of people improving in Alzheimer's disease, we are talking about the kind of getting all the family together at the dinner table at once kind of miracle, and not quite the walking on water miracle. So one story of somebody improving is not necessarily convincing. So you might say, wait a second, Jared. I've seen 25 stories of people getting better. So that is more convincing. Now it depends a little bit. Some of these clinics see thousands of patients. And so if you pick your 25 best patients, again, that's not surprising at all. But people have done random draws. So you take 25 random patients and they all get better. That is pretty impressive. That suggests that there is something going on here. But skeptics can still be a little concerned. Some of these, let's imagine your clinic is set high on a mountaintop and you charge $10,000 for every patient to come. You might be seeing patients who are very highly motivated and well-to-do. And maybe these patients are drawn from a population that was going to get better anyway. So the skeptics can still have their day. How do you address that? With a randomized clinical trial. So I've answered this question. So this is why we should do a clinical trial. But why does ISB want to do a clinical trial? Again, we actually want to do more than just test this hypothesis. We love testing this hypothesis. We think it's a great idea, but we want to do systems biology as well. So there's a couple other reasons we do clinical trials. One is to generate a lot of data so that we learn a tremendous amount about the disease that we're studying as we do the clinical trial. And one purpose of that, of course, is so that we can modify the intervention and improve it in a next version of a clinical trial. So we can do a series of clinical trials over time. And another reason is to give a gift to the research community. The more clinical trials we do on the more diseases, the more we can do integrative analysis across all these clinical trials to learn new things that we couldn't even imagine asking questions about before we started. And so that's sort of a separate question that I won't get into today, but it's a very important aspect of what we're doing research. We really want to take a global vision of human health as we're doing all these clinical trials and fit them together. So the COCO trial, as I've sort of alluded to before, focuses on telephonic coaching intervention as well as standard medical care, focusing on diet, stress, exercise, and brain training. Brain training involves doing these computer training computer training for the brain that focuses on sort of brain speed and attention. So it's not crossword puzzles, it's not Sudoku, but it is a way of engaging your mind. I suppose playing Fortnite would probably also work for those of you who are into that sort of thing. Where from a classic trial point of view, we're measuring the Mpi score, which is similar to the MoCA score. It's sort of how many items in a list you can remember over time to see if that degrades or improves as a result of the intervention. We're also looking at the FAST score, which is a measurement of function. So this is whether or not you can shop by yourself, whether or not you can get dressed and so on. Both are important, but fairly different measures of decline as a result of Alzheimer's disease. And we follow each of our patients over 2 years. Molly Raposa, whose picture is in the bottom, has been the main coach. She's been the face of the trial, or rather the voice of the trial, to most of to all of our patients, really for the last 2 years of the trial. We had additional coaches at the beginning of the trial, but Molly has really stood out for us in the last couple of years. A number of organizations have been involved in this, notably the Institute for Systems Biology, but also the Hoag Neuroinstitutes Institute in Orange County and the Schenkel Clinic, where the PI down at the Schenkel Clinic, being Rod Schenkel, has recruited most of our patients. The project has been funded from Providence St. Joseph Health. I want to acknowledge everybody before I run out of time. And then Rod Schenkel is the gentleman, and I just want to draw attention to him holding his glasses in the picture at the top of this slide. And also, I should say, probably the most important people to thank are the participants of the trial. The people who volunteer for clinical trials and engage in clinical trials are what make them possible, and are in some ways are medical heroes, not just for the COCO trial, but for any trial they participate in. So what makes ISB's clinical trials special? How is it that we're able to learn things that really normal clinical trials wouldn't even bother to try? And it's because for our clinical trials, we gather dense data on everybody, and we gather at multiple time points. We end up with these longitudinal time points. We throw out the phrase personalized, dynamic, dense data clouds, PD3 clouds. For COCO and for most of our trials, we get thousands of proteins and metabolites that we measure from the blood. We also measure dozens of clinical chemistries. And for COCO in particular, we measure a number of cognitive and functional tests. Together, that gives us about 2,500 variables that we're measuring over about 6 time points. Why are we going so big, right? So there's kind of 2 reasons that you can think of as it being important or valuable to get this much data. And I'm embracing both of them. I think both of them are great. And I don't want to be dismissive of either one. But one reason is sort of a fishing net. If I'm looking for one single fish in the sea that's really important, one of these analytes is particularly important to measure, I'm casting a very wide fishing net so that if it's out there, I'll find it. And the other reason is not so much that I'm looking for one, but I want to see the picture as a whole. So on the right here, we see a pointillist picture by George Seurat, an impressionist, and he's built up a picture that is beautiful. But if you drill in, it's each individual, it's points of paint that are here. He's built up a picture out of 2,500 or maybe a little bit more in this case to tell a story. So he's not looking to find a single point that's important. He's generating big data, using lots of data to tell a story and create a beautiful picture. So the fishing net is a little bit more like a 20th century paradigm for medicine. So doctors in the 20th century, out of the 19th century Johns Hopkins School of Medicine tradition, like to think of disease as being defined by single pathognomonic features, and later on that would be molecular features like biomarkers. And so we might think of like prostate cancer being characterized by PSA, for example, in the blood. So it may very well be that Alzheimer's is also driven by one or a few key things. And in all our humility, maybe we can't think of or imagine exactly what protein that is ahead of time. And so rather than spend 100 years testing each one at a time until a few thousand years from now we stumble across the right one, it makes sense to test as much as we can at once with a fishing net now. But that's sort of bioinformatics. Systems biology is really seeing the whole picture, putting all these data together as we measure and asking different types of questions. I'm going to try to give you a sense of how we do that with COCO in the next few slides, or in the rest of my talk, I guess. So as we put points together, if we gather very few data points, we get a very fuzzy picture, a very low resolution picture. Now, you can still do things with low resolution. So I could say, look, I have this picture and I'm a scientist, I want to do some kind of experiment. There's something about colors going on here, maybe red colors. So I'll let a cat loose and see over time if the red increases, right? So that's maybe an interesting experiment, at least intellectually, so it's not very satisfying. But the more points of data I add, it changes the nature of the questions I'm asking of the data. So when I have very few points and a very low resolution picture, I'm asking questions about sort of vagueness and colors. But when I have more points, I'm asking, does Polly want a cracker? Right. And it's not a slow qualitative difference. It's not a slow quantitative difference. There's just a change in the nature of knowledge. And statistically, it's kind of hard for me to say that there's a certain number of points at which point all of a sudden the data crystallizes and I'm asking different questions, the nature of epistemology changes. But each one of you can just look at the slide and say, yeah, there's a point at which my brain is thinking birds and there's a point at which it's not. And the nature of how knowledge is generated and the kinds of questions I ask differ. So with systems biology, we're not just trying to saturate carpet bomb tests, the so-called multiple testing explosion that you see in 20th century statistics. We're trying to use big data to paint pictures and answer different kinds of questions. Now, of course, I'm not doing pictures of birds. In general, what I'm doing is I'm using pictures of human interactomes. This is a small subset of the knowledge we have about the biological systems. This is a metabolic map. And so what I would like to do, what I'm aiming to do in one of the ways I might interpret data, is to paint all my data onto a picture like this and see how this picture changes over time and how it varies with people who are doing well versus not well, people who are in our intervention group versus our control group, and so on, and to learn from building up pictures like this. So this just reiterates the COCO timeline at 6 different points. We gather a lot of data. We also gather some data continuously, it's like Fitbit data for participants. Not everybody enrolls the trial at exactly the same time. So the first people started in January of 2018. The last person will finish up next week. So we actually started planning the trial about a year before this. It'll take us about a year to analyze the data. And for that matter, there'll probably be plenty of insights to be discovered by reanalyzing the data in years, many years to come. So we're talking like a 5-year effort to put this together. We had a couple of unfortunate events during the trial. So in 2019, Aravel, who was a company that we contracted most of our logistics to, dissolved. And so we had to scramble to put together a small team of people from our various nonprofits who were running the trial to take over their activities. And we succeeded in doing that. But we also stopped enrollment at that time in order to keep our task manageable. And then later, there was this worldwide COVID pandemic, which could have been disastrous. And in fact, for many clinical trials around the world, it was disastrous. We kind of got lucky because we were using a remote telephonic coaching intervention, which was just perfect in many ways during COVID. You might have said, well, maybe a bunch of people would drop out, and you can see that that's not actually what happened. In fact, if anything, I think people enjoyed having coaching, health coaching during the pandemic. And so that was the upside, is that one of the reasons we picked telephonic coaching and remote coaching is we really wanted this to be scalable, something that was useful, something that could reach out to millions of people. And so we've kind of serendipitously demonstrated that. A downside of the pandemic was for a period of several months, we were actually unable to gather some of the data that we were gathering in person, like blood draws. And so there's some delays or gaps in some of the data we collected. The upside is our data is so dense that we have analytical techniques that we can handle those issues. So now that we have all this data, or we will have this data soon, what do we do with it? I'd like to point out the trial's not over yet. So I can't tell you what the results of the trial are. I can kind of go on a flight of fancy, and I can share with you what data I have. But a lot of the molecular data we send off in batches. So even though we're only a week away from completing the trial, most of our molecular data we will not get until a week from now, when we send out all of the blood samples that we've accumulated for the last 2 years. I do have a little bit of molecular data from the first year of the trial. And so as a result, this flight of fancy, anything I show you could be wrong. I can change it next week. Please don't tweet it. And if you tell me I said it, I will deny it. But if we just look at the top level result, and I compare the primary outcome measures, so this is kind of the 20th century statistician analysis, not the systems biology analysis, we see that there's a difference between coaching and controls. It's significant. Why is it significant? Well, it may be that our intervention is actually working to improve cognition. And in fact, we see that cognition is improving. It's a non-zero slope. It's a positive slope. There are other interpretations that skeptics are welcome to use to explain these data. The one that is perhaps most interesting to me is the possibility that the coaching and control arms were not equivalent at the start. There's something innate about them. And during the randomization process wasn't equal, creating this difference. The nice thing about our dense data is, if that's true, we're likely to be able to figure out what that difference is and follow it with the molecular data. And so that is something that we can do with dense data that a lot of clinical trials would not be able to do if they had a result like this. The other thing that's sort of nice is that because we're measuring lots of different things, we have other pieces of data that are a bit orthogonal that kind of confirm this observation. So the FAST score, which is a measure of function, is also significantly improved in the coaching versus controls. We also see a number of molecular, if you want to call them biomarkers, that are correlated with these. And so there's a bit of a story that's coming out, and we'll see in our formal analysis when we start it whether this holds up or changes. Of course, this analysis is a little bit like looking at only one point of George Seurat's painting. I'm only looking at the Mpi score in the graph that I just showed you. So that's kind of looking like looking at like one pixel of this monkey's tail, the red pixel here, and trying to figure out whether or not everybody in the photo is happy, in the painting is happy or not. And maybe it's a good pixel to choose because if the monkey's waving his tail a lot, then okay, it's very indicative of the global story. But really, we want to come up with ways of looking at the entire picture. So with systems biology. So that means moving from sort of the low resolution upper left corner to the high resolution lower right corner and looking at the parrots and saying, you know, probably want a cracker. One way of doing that, there are many, many ways that we'll actually deploy. We have lots of different people with their own special expertises in systems biology and in looking at integrative data. But one way, in general, almost all of them though, use the combination of taking a large amount of past knowledge, everything that people have learned over the last century or more, particularly in the last 10 or 20 years, because knowledge is accumulating exponentially fast, and integrating that with what we've just learned, all of the data points we've learned from the COCO trial, and developing a picture. So Dan Fisher, whose image shows on the bottom left, is an intern from OHSU. He's just about to graduate with his master's degree, has been working with me for the last 5 months on developing these hierarchical edge bundling diagrams. George Seurat, eat your heart out, because these are beautiful. And what we're doing here is we're taking all of the measurements that we measure, or some subset of the measurements that we measure, and put them as circles on the outside of a circle, or little points on the outside of a circle. And if they're connected by some kind of statistic, so often correlations between the data that we see over time in the COCO study, we draw a line between. And then we start warping the lines to follow paths related to what previous knowledge will guide us. So that's a little bit vague, but let me see if I explain that a little bit better in this picture. I won't have time to explain it really well. So this is a hierarchical edge bundling diagram where what I've done is in black, I've drawn a tree of previous knowledge. So the black lines connecting all of the clinical measurements, the dozens of clinical measurements in the COCO trial, are nothing to do with the clinical trial. I pulled those out of 20th century knowledge. I could have drawn the black lines before I even started the trial. And then for all, this is only at baseline. So this has got nothing, so whatever we're seeing, the message that we're seeing from this picture has nothing to do with my interventions, just baseline characteristics on patients in the COCO trial. And then anytime two clinical chemistries are correlated with each other, I draw an orange line between them. And what we see here is that among the different functional groups, almost all of the clinical chemistries are correlated with each other within each group. But it's pretty rare for them to be correlated with each other across groups. But there's a couple that stand out. Notably, the glucose and the hemoglobin A1C markers, these endocrine markers, are connected with the immunologic markers like lymphocytes and neutrophils. So this implies that something to do with your diabetic health or your pre-diabetic health, or even for those people who are not diabetic, your sugar control influences the health of your immune system. So this is not surprising. This is known for diabetes, and but it's not necessarily something I would have looked at carefully in the COCO trial, because it doesn't immediately shout Alzheimer's disease when you think of those two concepts. And so this kind of diagram will allow us to monitor over time and visualize things like that are sort of emergent things that we may want to look at and paint more of an emergent picture. And of course, the hierarchical edge bundling diagrams are a bit of a step away from a full, highly resolved parrot picture. They're a step in that direction. And so we'll have other approaches where we map, such as metabolic modeling approaches, all of the COCO data onto pictures like this. But I'm going to leave you hanging there. I can talk more, but I know we're running out of time, not too much out of time, but I want to leave plenty of time for questions. And so let's see where we get, and maybe we'll come back to this during the question and answer period. Thank you.
Great, thanks, Jared. We have a really interesting question to get rolling in our Q&A. And just as a reminder, you can ask questions, use that Q&A box at the bottom of your screen to ask any question you'd like of Dr. Roach. And the first question is, with what frequency do you inadvertently enroll individuals with pseudo dementia such as those with undiagnosed hearing loss that can be corrected with hearing aids?
So it's not impossible that such a person could enter these trials. And indeed, in general, in people that enter the case studies where someone has Alzheimer's disease and they have a miraculous recovery, one of the key questions to ask is how good was the diagnosis, right? The miraculous recovery is the fact that they had some other treatable condition that wasn't Alzheimer's disease. So are we seeing any individuals in a trial like this? Most of the individuals in our trial, before they enter the trial, have had a year or more of being followed by a neurologist in an Alzheimer's disease clinical, in particular, Dr. Schenkel. And so tend to have really good diagnoses that are backed up with either PET scans or CSF biomarkers. As a result, I do not believe it's likely that any of the individuals in this particular trial, for example, had pseudodementia. But I suppose it's possible. We'll have some more data to look at that as we look carefully at all of the data in the trial, the molecular data.
Great, thanks. And I'll ask a follow-up question because we always get this question whenever we talk about trials and studies. How do people participate? How do these people get into the trial in the first place? And sometimes we have volunteers or people who would like to participate in a trial. Do you have any remarks for them?
So the logistics of most of our trials are such that they have to date required at least some component of in-person participation. And so it's made it very difficult for individuals who do not live near our trial site or sites to participate. And so the short answer is at our trial sites and near our trial sites, we use typical forms of advertising, flyers in the clinic, outreach to doctors who refer to the clinic, and in some cases, other forms of community advertising to alert people to our trials. Our trials are also all mentioned on the clinicaltrials.gov database. So people that troll that database, and it's publicly available from the US government, can sort of find out about our trials there. It's how I went and found out about COVID trials for kids to see if there are any in my area. It's a good database. Yeah. But the truth of the matter is ISB does not currently have like lots of big trials. So there's not ongoing opportunities for people to enroll in the trial. Our prevention trial, which is taking place in northern Los Angeles, in Santa Monica, at the PBHC, is still enrolling people with Alzheimer's. And that trial in particular, to address Dr. Laird's question earlier, requires a positive molecular biomarker for enrollment. So we're nearly certain we're getting people with classic research grade Alzheimer's disease. And there's a nuance between clinical and research Alzheimer's we can get into at some point if we want to enroll. So that's an opportunity. And then also ISB has a close affiliation with BHRI, the Brain Health Research Institute. We do not have trials there, but we are engaged in a longitudinal study with them. So it's not like we're randomizing people in an intervention or not, but anyone who joins that clinic has the option, it's free, to sign a consent form so that we can follow the molecular markers and the clinical trajectory over time in those patients. And then we'll be able to compare and contrast and contextualize those patients with data from our other clinical trials as well. But I suspect you could also follow the ISB webpage. We may announce these things on ISB webpage as well, our Twitter feed.
Sure, great. Next question. Could one outcome of the trial, once all data is analyzed, be a generalized intervention plan that people concerned about dementia could follow?
Sure. I try to follow all of the interventions that seem reasonable to me, which are most of the interventions. I was doing pretty well before COVID, and then I started eating all kinds of sweets. I fell off the wagon, and I don't have Alzheimer's. But the nice thing about our interventions is most of them have not only no side effect, no bad side effects, most of them are good to do anyway. So it's sort of motivating
Right.
to do these healthy behaviors anyway. But
Mhm.
there are some that take time or money, or maybe you're taking a supplement that could have side effects. And so it is reasonable to try to figure out which ones work for which people, to personalize these interventions. And that's definitely what we're trying to do with the data. At some level, in addition to sort of the top level averaged analyses that I sort of mostly talked about today, we're almost interested in ignoring everybody but one individual at a time in the trial and just studying the trajectory of that individual and what molecules are changing and tests are changing that individual to really understand what's going on with that individual. And we fully expect there to be sets of very different trajectories, people progressing for very different reasons in the trial, something that a typical trial would just never see, that just lumps everyone together.
Great. Okay, thanks. And you're not the only one who struggled during COVID to stay on some kind of regimen, Jared, I'm sure.
I'm gonna stop. I'm starting drinking coffee again too.
Our next question, can you describe what the coaching entailed and what the placebo version of the coaching was for the control group?
So this was not a blinded trial. It's very hard to blind recommendation to go out and get exercise. You know, so the fact that it's not blinded means that there could sort of be a placebo effect. We'll have to work through. A placebo effect's not the worst thing in the world, but it would be nice if it was more than a placebo effect, because placebo effects tend to fade over time.
Oh.
But generally speaking, placebo effects are not going to be reflected in molecular markers, and so we'll be able to tease that out. So the non-coaching receive standard medical care. They just continue to go to a high-quality Alzheimer's disease neurology clinic. They also received written recommendations. And as far as we can tell, the written recommendations, based actually on data from a prevention trial, have almost no effect. People tend to ignore those. So that's a placebo if you wish. The coaching was very similar, in fact, essentially identical to the kind of coaching that the Aravel was using for healthy coaching, for health coaching. So it was based on measuring clinical chemistries from individuals, and then the coach talking to somebody about what kind of recommendations they felt comfortable doing, what space they were at, and encouraging them to take a step up as appropriate, whether that was more exercise, reducing sugar from your diet, eating more healthy fruits and vegetables, and other sort of reducing stress, similar kinds of coach behaviors. And in addition to that, we asked everybody to spend about 2.5 hours a week doing brain training, computerized brain training. And those were just on the intervention, not on the controls. Now, at the end of the trial, all the controls got an opportunity, and many of them are currently doing this, to get 3 months of coaching. So many of them are actually getting coaching now.
Okay, great. Yeah. And now, I've heard you say this in Lee Hood, but there have been 400 failed clinical trials for drugs for Alzheimer's, and I know it's heartbreaking. For some people, I know we have foundation board members who've also served on the national board of Alzheimer's and the local chapter of Alzheimer's association, and it's absolutely heartbreaking to hope that one of these drugs is going to work and then they fail again. Do you have any comments about that?
Well, kudos to the Alzheimer's Association, because increasingly, I've just noticed over the last, I don't know what the right time frame is, but maybe 2 or 3 years, it's probably longer. They're really changing their emphasis a lot and recognizing that indeed the ideas for treatment of Alzheimer's have blossomed. But the vast majority of money for testing drugs in the United States and the world comes from big pharma, right? They are not going to test non-pharmacological approaches to treat Alzheimer's. There's no money in it for them. So they're going to test things that they may have a chance of making money on.
Right.
You don't really need me to comment. You can Google this. There's lots of articles that talk about this. I found there's one where it sort of says, the definition of insanity is doing the same thing over and over again and failing. And if that's the definition of insanity, Alzheimer's research is insane. There's, if you kind of want to argue the opposite point of view. There's some suggestive evidence that maybe some of these newer approaches, like this, if you follow the aducanumab story, may have a mild effect. So you could argue they're not complete failures, but they certainly succeeded in disproving the amyloid hypothesis as the only cause of Alzheimer's disease, for sure.
Okay. Okay, thank you. Have you compared remote evaluations of cognition that have only audio exchange versus audio plus visual, as in a Zoom or a FaceTime meeting? So the question is, does the visual information slow, degrade, or improve the cognitive responses in your patients?
Okay, it sounds to me like there's some really interesting insight behind this question that Charles has my email address. He can email me or call me later.
Okay.
But the short answer is not in this trial. A slightly longer answer is we've had difficulty coming up with all remote evaluations of cognition. We have several options available to us. BrainHQ has some, there's others that are being developed. Some of our psychometricians who have been delivering some of the traditional measures through remote Zoom thing. Our problem is that at least when we're in the realm of communicating with longtime Alzheimer's disease researchers, they like these things called validated tests. So tests that have been done again and again in lots of different clinical trials where they say, we know there's a response. We know that there's certain types of statistical behaviors to these tests. And unfortunately, almost everything online isn't validated. So although we can do them, it means that when we turn around and try to write papers and present those results, which we're, to be honest with you, we're going to have to do with some of our studies that we're doing currently in the future, we will have to overcome that burden. And so, yeah, I'm interested in any ideas and data and recommendations. Thanks for the question.
Yeah. Okay, and then the last question, and this is a really great one to end on. What is your assessment of the current standard medical care for those diagnosed with Alzheimer's? And if a loved one were diagnosed, how would your recommendation to them differ or not from the standard care?
I want to be very careful about suggesting anything that's generalized. You know, I think every situation is different. So for example, I look at some of the pharmacologic approaches. Most of the pharmacologic approaches, at least some of them, tend to have at least brief bumps in cognition or function for up to a year, maybe a little bit longer. And then often there's sort of a steeper drop off and they end up back to where everyone else is. I don't think that would be that important for me. I don't think, but maybe there's certain situations where you say, you know what, I want to try to improve things for a year or two, and then it's okay if I fall off the cliff. I would really double down on as much as I could on lifestyle changes, right? Can I improve my diet? And there's detail. It's not just any old diet, but can I decrease the amount of sugar I'm taking? Can I get more exercise? Can I learn new things? So cognitive stimulation, learning a new language. It turns out doing the same old things like crossword puzzle in your life doesn't seem to have much of an effect. It's really expanding your brain. And some of these brain training tools, like from BrainHQ, which is the company that is collaborating with us to provide brain training, I think are useful and I'd recommend. I would pay attention to research, even the drug company research. Just because you try 400 times doesn't mean there won't be some progress. I look at this, the aducanumab story. When I look at the data, it seems like the side effects outweigh any possible benefit, but who knows? Maybe they'll be able to tweak that. And it may depend on who you are, right? Personalized interventions may matter, whether or not you have, like if you have a known rare genetic cause of Alzheimer's, there may be some drugs, like if you're from this region of Colombia, for example, where a PSEN1 mutation is common, there may be some opportunities to have very personalized care. Okay. I'm happy to keep talking, but maybe our time is up.
Yeah, we're pretty close to having our time up, but it's a good problem. That was a nice, robust conversation. Thanks, Jared. So
Tell you what.
Yes, what?
Okay, go ahead. You're welcome. I was going to say one more thing, but we'll
ahead,
leave
go
that
ahead.
for later.
Go ahead.
All right, here's my coffee story.
Okay.
So one of the things that I thought is kind of interesting is how we connect information and paint our picture. And so what I can do is look at all of the known metabolites of caffeine, and that's metabolized in the liver, in our metabolomic data. So it's kind of cool. So I can take, even though it's not my intent to study caffeine in the cocoa patients, I can. So what does it look like? If I look at caffeine and all its derivatives, so first of all, the derivatives of caffeine, which caffeine is metabolized in the liver into things like theophylline, theobromine, they're all almost perfectly correlated with each other. And then they're all fairly well correlated with caffeine. And so you can see we have a variety of caffeine drinkers in the cocoa study. And it tells me something about these particular individuals. Now, I did try to take a look at the levels of caffeine and whether or not there were cognitive changes, and I didn't see anything. So there's probably no story here about improving Alzheimer's disease, but there is a story about connecting all kinds of metabolites and seeing a picture in individuals. And there's actually an even greater picture that shows up here, because everybody in this study was told not to drink any coffee before they went in for their blood tests. So I may not actually be measuring caffeine intake so much as I am measuring sort of the free spirit nature of the participants. And to those people, if there's anybody here, I'd like to point out I participated in a similar study and was told not to drink coffee, and I did. So I'm one of those outliers. So this is fun. This is the things you can do with data. And it's an example of why generating dense data is inherently valuable in ways that are not predicted, because there's so many little stories like this that will come out of our data that are not only related to Alzheimer's disease research, but many other aspects of biomedical research. And by combining these little stories and insights across all of ISB studies, I think you're going to see a lot of emergent knowledge that just would never come if we didn't do studies like this. All right. I got my little caffeine story in. Thanks so much.
That was great. And you know what? I loved your picture of the parrots too. I thought that was great in terms of more and more data and the clearer picture. I thought that was a great way to position it. Good way to explain it.
Okay.
So yeah, thanks, Jared. And okay, and thank you all for joining us today. And we do have 2 more events coming up in June, and we'd love to see you at both of those. On Thursday, June 10th at 6 p.m., we're partnering with Town Hall Seattle to bring in Dr. Temple Grandin. She's a prominent scientist, and this is her new book, as we pointed out, and also our giveaway book. She'll be in conversation with Jim Heath, and you can get your tickets at the Town Hall Seattle website. You can also go to our website, isbscience.org, and go to our events tab, and we'll give you a link to get over there and get your tickets. And we'll announce the winner of this in just a minute. Our second event is our annual STEM education fundraiser, and that's on Thursday, June 17th at noon. And our education team has just worked overtime this past year to pivot and really take care of our teachers and school districts in a way that was, it was heroic, the efforts they put in. I can't tell you how hard they are working. And we would love to see you all this event. And hopefully we'll inspire you to make a gift to our STEM education efforts there, but you're all welcome. And you're on our email list, so you will get more information about that. And the winner of our book tonight is randomly generated, was Ron Subert. So our friend Ron, I will figure out a way to get this book to you. I'll connect with you afterwards. Congratulations, and thanks for joining us today. And that is all we have. Thank you, everyone, and hopefully we'll see you in June. Bye-bye.